So what specifically do you not understand about cell membranes?
Well, I understand the cell membrane. It allows things to travel in and out of the cell, to let it in and out. And there's different proteins that you have the integral and the peripheral. Let's just get to the basics. We talk about the phospholipid layers, right?
Okay.
So you have the phospholipid heads, which are hydrophilic.
Yep.
They just water loving. And the hydrophobic tails, which are Is it recording or not?
Yeah, it's supposed to be.
Oh, cool.
See the heads, which are what?
Hydrophilic, water loving.
The tails, which are
Hydrophobic.
And you know what these are?
Those are phospholipid.
Yeah, they're phosphate.
Phosphate.
You know what the tails are made of?
No.
Fatty acids.
Fatty acids, okay. I knew there's cholesterol and stuff in there. Does that have anything to do with it? No, the tails are just fatty. Okay, yeah, because okay.
The cholesterol basically sits kind of like this.
Mm-hmm.
And it just gives the membrane its structure. It helps keep it so can't move too much, because it needs to be the fluid mosaic model. Have you heard that term before?
Yes,
You
definitely.
know what that means?
The fluid mosaic model, I don't know the exact definition. No.
It's you don't have to know the exact definition. It's just the ability of the cell to change.
Okay.
It's constantly moving, constantly changing ions and glucose and nutrients, everything always coming in and out, gases, everything. So it's just the ability of it to continue to kind of change and adapt and bring in stuff it needs, get rid of stuff it doesn't need, all that kind of stuff. That's all
Okay.
the fluid mosaic model means.
That makes sense.
So that's what cholesterol does. Cholesterol just gives it a structure.
Mhm.
Just helps keep it, gives it more structure than
Exactly.
just having these. And then, so you have the protein channels in between.
Yeah.
And those act to
To be an integral.
bring stuff in, get stuff out. So
Okay.
one of the ones you have is your sodium. Whoops.
Is this what I'm going to be learning next in anatomy?
Yeah. So this will be like a little prequel for you.
That'd be nice to have a.
So this is the sodium potassium pump. So it's constantly pumping sodium out, and potassium in.
Okay.
Another important thing to know about the cell is that on the outside, there's a high concentration of sodium.
Okay.
On the inside, do you know what there's a high concentration of?
Potassium.
Potassium.
Mm-hmm.
And this creates the gradient. So when you cause when an action potential starts,
Yeah.
that's what makes the sodium want to rush into the cell.
Yeah.
Because of
It goes from a higher grade to a lower?
Yes.
It's trying to go to a lower grade, okay.
Yes. Stuff always wants to move from higher concentration to lower concentration.
Okay. All right.
So that's why it's important for us to have this difference, so that when and then so over here, let's just an example. You have
What type of channel?
kind of look like this,
Yeah.
which is a
Is that gated?
sodium voltage gated.
Okay.
So this does is when, have you learned about this graph right here?
No.
No?
That's for next week.
Oh, that's for next week? Okay.
Yeah.
Let's not.
We talked briefly about it actually in class today. We talked a little bit about the.
Well, the important thing to know is
Yeah.
when sodium comes into the cell,
Mm-hmm.
that's what the action potential is.
Okay.
Does that make sense?
When sodium comes in, that's what the action.
So on your little graph of action potential, when you have your threshold, you're coming up, this is stuff entering the cell. Well, the voltage gated channels aren't activated yet. That stuff coming into the cell, and then once it goes up and reaches threshold, it causes action potential. All of these voltage gated channels are going to open. So it has tons of channels on the outside, like 50,000 protein channels on each cell.
Okay.
Like a lot.
Wow.
Protein.
They're negatively charged, right? Proteins? Or you're talking about channels?
Proteins aren't. Yeah. Proteins aren't charged.
Okay.
But the cell membrane is about 50% phospholipid heads, fats, tails, and cholesterol, and then 50% protein channels. That's how many protein channels are. They make about half of the cell membrane.
Okay.
So I don't know if there's anything more specific on your study guides.
Yeah, there just talks about we talked about the different proteins, you know, the peripheral, the integral, and what their functions are. And then role of carbohydrates in the cell membranes.
Role of carbs? I don't know if that's just talking about
Where do carbohydrates look?
secondary active transport. Will you have any more specific questions about the cell membrane?
I don't think so. I mean, that's the basics of what
Yeah,
I
it's
need
pretty-
to know right there.
Yeah.
You have sodium on the outside, potassium on the inside, and that flow there. So you have sodium flowing in, and you have potassium flowing out.
Opposite.
What's that?
It's constantly kicking potassium or sodium out.
It's kicking sodium out.
Yeah, 3 sodiums out, 2 potassiums in.
Okay. So that's why.
And that's creating this
it's where they're
gradient.
at. Okay. Yeah.
Yeah.
Okay. Gotcha. All right. That's cool. He started to talk a little bit about membrane potential. So we talked about unequal distribution of ions because of the. It kind of had to do with. What is it? Establishes membrane potential. So you have the sodium going out.
Yeah, that's what we just talked about.
That's what we just talked about, yeah.
You can create the graded potential.
Create the gradient potential, okay.
So there's higher concentration of sodium outside, higher concentration of potassium
Okay,
inside.
and then it's trying to reach equilibrium, right? Is that what it's trying to do?
Yeah, you also have these channels where you're just leaky,
Yeah, exactly. Just open.
Potassium
free flowing.
channels?
Yeah,
which they just let potassium leave on its own. So that's what's going to help keep the cell
yeah.
resting at, I believe, the cell
Minus-
heats at negative 55.
Well, maybe the lowest is negative 70, but it's negative 55,
Oh, so you guys are talking about negative 70 right now?
negative 70, yeah.
Okay, that's the
Which
number.
that's the lowest it can go, right?
No, it goes a little bit lower during action potential because it goes too far down, it comes back up. But
Okay. Was that resting?
We won't jump too far. Yeah, so resting membrane potential is rest at negative 70.
Okay.
And that negative 70 is allowed to sit right there because of the sodium potassium pump and the leaky potassium channels.
Okay.
I believe, I remember having a test question about that, and I believe it's mostly this guy right here. Is what helps maintain the negative 70.
The leaky
Mhm.
potassium pump?
Not the pump, just the leaky channel.
The leaky channel, okay.
And it's always open and just lets potassium go out freely.
Okay. All right. I'm trying to think if there's anything else. I had a couple of questions written down.